Today we are opening the morning sessions with Professor Helga Stoer from VIBR University, and she's going to talk about micronuclei.
Good morning. Thank you for the introduction. Okay, so micronuclei, what are they? Micronuclei are formed from either acentric chromosomal fragments or from whole chromosomes during mitosis. So when in the cell a chromosome break occurs, the chromosome fragment sits there until mitosis, and then after mitosis, when the new nuclei are formed and a new nuclear envelope is formed, then these little pieces are enclosed by a separate membrane to form these little structures. It could also be a whole chromosome that lies apart because maybe the spindle of the mitosis is disturbed. And this is what it looks like in the microscope. You have the normal nucleus and you have a little micronucleus. And by the way, if you have questions, you can interrupt me at any time. I think we can rather skip some of the more boring slides and instead get a discussion going if you like. So, interrupt me at any time. So what can we use micronuclei for? There's several things we can do with them. It's a mutagenicity endpoint, so a genotoxicity endpoint. It's not, for other toxicities. So, but in that it can be used for a couple of different things. First of all, there is a mutagenicity testing of chemicals in vivo. So if you want to admit a new pharmaceutical, for example, you have to supply a lot of data, and among them usually an in vivo mutagenicity test, which can be the mammalian erythrocyte micronucleus test. It's done usually in mice and or rats, and it uses reticulocytes from the bone marrow that can be assessed microscopically or by flow cytometry. Then there's, for example, the hen's egg micronucleus test, and I'll have a little explanation for each of them. And then there is what I want to talk most about today. This is the mutagenicity testing of chemicals in vitro. So the mammalian cell micronucleus test, and there's an OECD test light guide for this one. There's also one for this, but this is my main emphasis today. And then you can you can use the micronucleus test for looking at environmental pollution. For example, you can assess cells of environmentally burdened plants or animals of different kinds, and I have some examples later. And also you can do human biomonitoring related to exposures or diseases. You can do it in, for example, human lymphocytes or in buccal cells, or you can use some other cells. And this is Stefano's topic afterwards. So the in vivo micronucleus test, I just want to cover it very briefly. So do you have a rough idea what it is? You use the bone marrow cells of treated mice or rats, and what are analyzed are reticulocytes, which are young erythrocytes. And the thing is that during maturation in the bone marrow, the reticulocytes expel the nucleus. But for an unknown reason, they keep the micronucleus. So what you have afterwards is these young reticulocytes, which only have the little micronucleus. So if you analyze a lot of reticulocytes, there's no DNA in most of them, and just in very few of them, there's this little micronucleus. So it's very simple to see this, and it can be done. It can be done either by counting bone marrow smears with the microscope by eye, or it can be done with flow cytometry, which is then, of course, much less work. And you can then use peripheral blood samples and analyze for those young reticulocytes. And then there's the hen's egg test. This is something, well, not fairly new, but it has gained increasing acceptance recently. What is done essentially is really you take eggs and you breed them, you put a little hole and you put the compound into the hole, the test compound, and first you breed it for about a week, 8 days, and then you put the test compound, you leave it for maybe a day or two, and then you cut the egg open, you take out a blood vessel, and you just put the reticulocyte, the erythrocytes, so the erythrocytes on a slide. And it looks like this, and because hens are birds, the reticulocytes have a nucleus, and here you can see a little micronucleus. This is an example of what it might look like. If you look at the data, there's mannitol, a negative control, and then there's ethylmethane sulfonate, a positive control. And you can see here the micronucleus rate, and the negative control is negative, and the positive control shows a dose response type curve. The advantage of the Hens egg micronucleus test is that there's metabolism going on. It's also something like a complete organism type thing where you have distributions and different compartments. And so that's why it's gaining increasing acceptance. Here are some other examples where they have tested MMS as a positive control, and then they have tested a pesticide, which was positive, and another pesticide here, which was negative in this test. Then the in vitro micronucleus test. So which cell types can we use? I said it's a mammalian cell mutagenicity test. We can use pretty much every cultured cell line as long as it's, the nuclei look reasonably round and they have a stable karyotype. And good examples for that are TK6 human lymphoblastoid cells. That's actually, I think, nowadays the most often used cell line. Another good example are mouse lymphoma cells with this name, L5178Y, and they were used because there's an established gene mutation assay with them. So for a while, they were very popular for in vitro micronucleus testing. And even before that, but still sometimes today, people use the V79 Chinese hamster cells. And that was because there was an established gene mutation test with them. So you could in parallel assess micronuclei and had different endpoints in the same cell type. And you can use, for example, HepG2 human liver cells, which have some limited metabolism. So you may not have to add S9 mix, which you have to do for the other cell lines because they cannot metabolize the compounds usually. You can also use peripheral blood lymphocytes for testing chemicals. And you usually need to add a metabolic activation extract for red liver extract to enable the system to metabolize the compounds. So there's a specific thing about the micronuclei in vitro. You can use cytokeratin B. And what does it do? Cytokeratin B is a cytokinesis inhibitor. So we remember this picture here where just a single nucleus in a new cell has a micronucleus. But what happens is that the cell goes through mitosis, and usually you get these single cells, and one of them may have a micronucleus. When you add a cytokinesis inhibitor like cytokeratin B, the nuclei still separate, but the cell does not separate. So you have one big new cell with two nuclei in it. And the nice thing is about that is that you know that this cell has undergone one mitosis since you have applied the chemical in question, the test chemical. You apply it and you're sure the cell has undergone one mitosis to express the micronucleus, not more, not less. And what it looks like then is that you have these binucleated cells with a little nucleus, or even two micronuclei or more sometimes. So some more just general remarks about the in vitro micronucleus test and what's special about it, what you can do with it, what you can see. So micronuclei represent damage that has been transmitted to a daughter cell. So that's somewhat heritable damage, which is, some people at least think, more meaningful than if you assess damage that has not been inherited to daughter cells. Then it detects aniogens and clastogens. So it detects compounds that cause the loss of whole chromosomes, which are aniogens, and clastogens, which are chemicals that cause breakage of the chromosome. It can be done with or without cytokeratin B. We've just covered that. You can also do it without, but then you have to use a good cell proliferation assay in parallel with the micronucleus test, because you have to make sure that your cells are still going to be able to go through mitosis. Otherwise, a micronucleus test is not meaningful, because mitosis is needed to express the micronuclei from the damage, right? Before mitosis, we just have the chromosome piece, but we don't see it. Only after mitosis, after it has been enclosed in this separate membrane, we can see it and count it. And that's the nice thing about cytokeratin B, that you have the automatic inbuilt reassurance that there was one mitosis. And then when you count the cells with one nucleus and two nuclei and three or more, you can also calculate a proliferation index. And because they have three nuclei or four maybe, then they have undergone a second mitosis already. If there's only one nucleus, no mitosis. So you can count an index, and that shows you very nicely the proliferation ability of the cell culture. You can also see apoptosis on the slides, which is also a nice additional thing. And here I want to show the value of the proliferation index. Here you can see in TK6 cells on the right side, you can see results after treatment and the doxorubicin in this case, doxorubicin here. And you can see that if you look at the viability, which we did in parallel in a separate experiment, viability goes down with higher doses a little bit, and the highest dose is really down considerably, but the others are not very much changed. But when you look at the proliferation index, which is this one, you can see very clearly an earlier, you can earlier with the lower dose see the effect very strongly. So in a lot of cases, the proliferation index is more sensitive than a typical viability assay, which is an assay for live and dead cells, basically. That's the point I wanted to make with this slide. And this is about the proliferation index. So how is it calculated? It's the number of mononucleated cells plus 2 times the number of binucleated cells plus 3 times the number of multinucleated cells divided by the total number of cells. And what you end up with numbers like between 1 or 1.5 and 2 or 2.2 or something like that, in that range. What does it mean? For example, here you have a proliferation index of 2.1. And that is that you have a low number of mononucleated cells, a very high number, like 700 of 1,000. This is counted in 1,000 cells. So 700 cells are binucleated, and a few cells have undergone another mitosis in one of the nuclei. So there have three or more nuclei. So this is an ideal picture where you have most of the cells in the state that you want them in. That results in a proliferation index, CVPI, of 2.1. Here's another example where you have half of the cells as mononucleated cells, and you have 400, a little bit less than half, as binucleated cells, and you have 100 multinucleated cells. Now you can still analyze this because 400 cells is still enough, so that by adding more cells, you can reach 1,000 binucleated cells to count micronuclei. Still enough, but you know that your treatment already inhibited proliferation a bit, or that the cell culture is not good. That depends on, you have to compare with the control and the treatment, of course. But then you have a CVPI of about 1.6. So I said you can detect apoptosis, and this is what it looks like. So this is an apoptotic cell, here an apoptotic cell, here an apoptotic cell. If you have cells that grow adherent to the cell culture, you may lose some of the apoptotic cells because they detach, and you wash the cell culture before you prepare it for micronucleus evaluation. So you may lose the detached cells, but you still have some apoptotic cells in there usually. If you have a suspension cell culture, you don't lose any cells, so you have all of your apoptotic cells still in the culture. So not in every case can you use this assay to assess apoptosis quantitatively, but you see whether it's there. If you have a strong or even some apoptotic response, you will see it because there will be a few more on the slide. So that gives you a mechanistic indication for a certain mechanism of action of the compound that it causes apoptosis. On the other side, if you have a lot of these things there in the culture, you may not be able to count micronuclei anymore, because if you have a little later stage of these cells, the apoptotic bodies are not kept together by a membrane anymore. They might lie on top of the other cells somewhere, and if you have this little thing lying on top here somewhere, you might find it hard to distinguish it from a micronucleus. So if you have a huge number of apoptotic cells, you may not be able to do the micronucleus test under these conditions. So you can do another thing with micronuclei as well. You can see what is in them. You can stain them with either immunofluorescence, an antibody staining, or with fluorescence in situ hybridization for the centromeric region of the chromosome. There are antibodies which stain the proteins around the centromeric region. Question? Yes, please.
The micronucleus test with flow cytometry, but can you detect the dying cells easily with flow cytometry too?
Well, that's actually an excellent question. The flow cytometry I introduced for the in vivo micronucleus test, and there you don't have that problem. But you can also do the in vitro micronucleus test with flow cytometry, and then you have exactly that problem to distinguish from apoptosis. People started trying to do that decades ago with flow cytometry, and then there were only a few experts who were able to distinguish apoptosis from micronuclei. However, they have now modified the procedure, and at least if you use certain kits that you can buy and you establish the method carefully, then you can use flow cytometry for mammalian cells. It is then easiest with suspension cell cultures, of course, but you can do it, but you need good knowledge, you need the kit to do it, and you need some time to set it up and a suitable flow cytometer. But then it's true, that's exactly one of the issues that they had for a long time. Okay, kinetochore staining or centromere staining, what is it good for? You detect the centromeric region of a chromosome, either the proteins that are there or the DNA that's there, the proteins with the antibody staining or the DNA with the fluorescence in situ hybridization. This is a picture of immune fluorescence staining, so with antibodies. And here you see micronuclei, and here, this is the same picture. You see that there's no signal dot in them. Here you have the micronucleus, you have a signal dot in it. And so that means that the centromeric region of a chromosome is in the micronucleus. Now, that could be a sign that you have a whole chromosome there or the centromeric fragment. If you have the signal in a majority of the micronuclei, you tend to think that you have whole chromosomes in your micronuclei. That means that the mechanism of induction of micronuclei by this compound is mitotic disturbance. Here's an example for that, what it might look like data-wise. You can treat with mitotic spindle disturbing compounds, for example, colcimid, diacylstilbestrol, it's a synthetic estrogen, which causes spindle disturbance, griseofulvine and vinblastine, two more spindle disturbing substances. And you can see that you get micronucleus induction. And if you only look at the micronuclei and look how many of them have a kinetochore signal, you can see that you have over 80%, in some cases over 90% positive micronuclei, and the control in this case only had 20% positive micronuclei. This is from a cell line, and some cell lines have like close to 50% or human lymphocytes, but the cell line has only 20% positive. So you see a huge difference, and from that huge difference, you can conclude that you have a compound which causes mitotic disturbance and whole chromosomes to be put into micronuclei. If you want to be sure it's a whole chromosome, you have to do a combined centromere and telomere staining. That can also be done, but usually it is for determining the mechanism, it will be enough to do this. There's an OECD guideline that tells you how to do the in vitro micronucleus test. The OECD guideline, of course, is mostly for industry who wants to develop substances and to gain the data for submission for approval of the substance. But it's not exactly what we do in research, in basic research at university labs, but pretty close, and you can use it as a guideline how to do a proper micronucleus test. So what does it say? I want to just pick out some very important points. It says first the cell lines are either cell lines or primary rodent or human cell cultures, and they have to have a stable and low background micronucleus frequency, and then all of them are suitable. So it's not limited to what just a couple of cell lines. You can use any cell line if you can prove that it has a reasonably stable and low background micronucleus frequency. Usually, a metabolic activation system is required. You have to add S9 mix to have a complete test. And you use normal non-synchronized cells usually that are in exponential growth phase. They have to be in exponential growth phase so that they can go through mitosis and express the micronucleus. And you need a solvent control, and you need at least 3 test item concentrations to have a good experiment. And usually when we do research at a university lab, we need at least 3 independent repeat experiments. Now, for admission of a compound for regulatory purposes, you do not need that. You have no requirement for verification of a clear positive or negative response. But that's said in the guideline. But remember, before the industry submits the data for regulatory purposes, they have done many experiments with the compound. Just the final guideline conform the the experiments according to guideline that they then submit has to be just 1 experiment. So what else is in the guideline? For example, there's a recommendation about the substance concentrations that you should use. You should go up to substance precipitation or cytotoxicity, or there are certain upper dose limits. If you don't reach any cytotoxicity and the compound is still soluble, then you can stop when you've tested up to this pretty big amount of compound. And you should cover the concentrations between cytotoxicity and also no cytotoxicity. And cytotoxicity is defined in this guideline, and in this case, it's a reduction of the CBPI, the proliferation index, to 45 plus minus 5% of the control. If the compound is only positive at a toxic concentration, then you have something what people like to call a high toxicity genotoxin. That means you can discuss the relevance of the data. Is it really relevant? I mean, does this effect occur? Can it occur in vivo in humans? Or is it something that can only occur in cell culture because it's associated with so much toxicity? You could not reach that concentration, for example, in humans. Then you can start discussing that. And you need to add a suitable positive control, of course. And the positive controls are also listed in the guideline. And there are positive controls that are recommended for testing substances where you do not expect that they need metabolic activation. And then there's methylmethane sulfonate, there's mitomycin C, there's NQO or cytosine arabinoside. Then you need a positive control if you think your compound might need metabolic activation. And the typical ones are benzoylpyrene and cyclophosphamide. And if you think your compound might be an antigen, it might make sense to add a positive control of that category. And colchicine or vinblastine are the examples that are given. So they have some more information about how you should perform the test, which are useful if you also, for university type research as well. You should treat for 3 to 6 hours, then remove the test substance, and then harvest after another 1.5 to 2 cell cycle durations. And that is because if you have a compound which causes arrest in mitosis, if you treat it for the complete time, the cells will never leave mitosis and not form a micronucleus. So it's better to treat it for just treat the cells for just a limited amount of time, then remove the test compound, and then they can start going through mitosis and form the micronucleus. And to be sure that they were able to do this, you leave them time 1.5 or 2 cell cycle durations. If they are negative, if it's negative, the test result, you can treat for a longer time and try that. But that only makes sense without S9 mix, because S9 mix usually is toxic and you have to apply it for a very long time. And it also says in the guideline that you can modify this if needed and if you have good reasons for it. Importantly, the slides are coded before evaluation. This is really a requirement. Even in basic research, you need to code the slides or have someone else code the slides better than code them yourself. And you have to analyze the micronucleus frequency in at least 2,000 cells. So usually you count 1,000 from one slide and another 1,000 from another slide. And you can collect your data for historical positive or negative control. Historical control means that you accumulate the control values over a certain amount of time that you have worked with the same system, for example, the same cell line and the same type of incubation, duration, and treatment, and so on. You have a series of experiments where you expect it takes maybe half a year or a year, then you can collect your positive control data and your negative control data and establish a historical control. For that enables you, if you have a particular experiment and you have a bit strange results, to see whether it falls in the normal range or not. And if it's not in the normal range, for example, the control on that day was double of what it usually is in the historical negative control, then you can say, okay, something was wrong with my culture. I'm not going to accept this result. So what else can the micronucleus test tell us? It can tell us even more. You can assess, for example, the occurrence of so-called bridges, chromatin bridges. And chromatin bridges, they can form because of certain repair events or incomplete chromatid separation or telomere end function. For example, inhibition of DNA methylation of topoisomerase II. So some mechanisms can cause problems with chromosome separation, chromatid separate the mitosis, and then you see this little bridge here. This is an actual microscopical picture, what it looks like. This is the scheme down here. It can be in addition to a micronucleus or without one, and it can give you an additional hint about the mechanism of action of the compound. What may be also important to mention is that DNA repair capacity can influence the results in the micronucleus test. These are results, they are old results, but it shows that difference nicely. These are control cells, the lower solid line, and here also control cells, and these are cells derived from those, but with a repair deficiency. And you can see that the induction of micronuclei is not only starts at a higher level, but it's also, the cell line is more sensitive, so the line goes up steeper. Here also the same. So if you have repair deficiency, at least some types of repair deficiency can lead to an increased sensitivity in the micronucleus test. Now the question is, now these micronuclei containing cells, what happens with them? Where do they go? Do they just stay around? Do they form daughter cells with micronuclei or without micronuclei? And why is that even important? Well, it is important because if they die, then they cannot form a transformed cell, a tumor. You know, we are doing this genotoxicity testing to identify substances that pose a carcinogenic risk because they are mutagenic. So we're interested in the question whether this compound could possibly cause a tumor formation in humans. Now, if it only causes dead cells, it will never cause a tumor formation. However, mutated stem cells, for example, they may form a tumor. No, dead cells do not form a tumor, but mutated stem cells may form a tumor. So it is really important to know what the fate of a micronucleus containing cell is. And we have early on asked the question, could the loss of tumor suppressor genes through micronuclei, could they cause cell transformation? For example, here in this scheme, you have a micronucleus, and let's assume a tumor suppressor gene is in the micronucleus. Now, if you lose that micronucleus, would you have a transformed cell? And we were trying to see that in a model system, which was a gene mutation assay, the mouse lymphoma mutation assay, and it's supposed to be sensitive also for chromosome mutations. So that means if the selectable gene in that assay, if that ends up in the micronucleus, and then the micronucleus is lost, you would expect to get a mutant. So we did this assay, and you saw these results before. We have a lot of whole chromosomes in the micronuclei, and we did mutation assays, and we calculated what the mutation frequency should be. And there was no significant increase in the mutation assay with these compounds that cause the loss of whole chromosomes, even though the positive control was okay. But we thought, okay, it might be that the one chromosome that we are looking for that has this selectable gene does not end up in micronuclei for some reason that we don't know. So we did chromosome 11 specific painting of micronuclei. That's the chromosome where that gene in question lies. And we did that, and we looked at micronuclei, and we calculated the frequency from that we would expect, based on the frequency of chromosomes that are positive for this chromosome. based on the frequency of micronuclei that are positive for this chromosome. We analyzed the mutation frequency that we would expect. And we would expect a 4-fold increase in mutation frequency, but we did not get any increase. So that does not seem to happen. It does not seem to be possible in this mutation assay, at least, that you lose a whole chromosome by micronucleus formation. As a result of all these questions, we formed a group, a working group within the GUM, the German Mutation Society, Gesellschaft für Umweltmutationsforschung, so the German-speaking subgroup of the European Environmental Mutagenesis and Genomics Society. And it was a working group. We worked several years and came up with this paper summarizing the fate of micronuclei and micronucleated cells, what is known about them in the literature. And these are the possible fates here. So what you could have is that the micronucleus could just reintegrate with the nucleus. And then it may be, if it reintegrates well, that you have no mutation as a result. It could be that the micronucleus is destroyed by the cell because it doesn't like that extra material and has mechanisms to get rid of it. Then if it's just really lost, there might be a mutation possible as a result. If genetic material is lost, that the cell still survives. There might be persistence. It might just stay there. And then you may not have a mutation if it's still active genomic material. These are just the theoretical considerations now. Or you might have apoptosis because this extra thing might be a signal for the cell to go into self-destruction. And then you lose the whole cell. You have no mutation. Or you might have chromotrypsis, and this is the most recent findings that people have reported a few years ago. That means that the material in the micronucleus is rearranged and then it's reintegrated into the main nucleus. And because it's rearranged, it's a mutation, or it is mutated, it is highly mutated and might cause a mutated cell. Yes, sure.
Will possibly lead to a mutation, while persistence may not lead to mutations, because is then the micronucleus still used to transcribe, for example, proteins? Because I would suspect that we go to the main nucleus for our transcription.
Very good question. Yes, excellent question. Well, these are just hypothetical considerations now, and it means that if the micronucleus is destroyed and if an important gene was on there, then that's lost, and so it's a deletion of a gene, it's a mutation. Here, it is, if you think it would have no mutation, you're totally right. You would have to assume that the material is still active and can be just transcribed. There is research on this part also in the literature, and some micronuclei seem to be accessible and some aren't. It depends on the nature of the micronucleus and probably the cell line. And here's an example. And I think this was supposed to be a movie, but it's not, so we leave it out. So here we have a paper in the literature which dealt about the question of dealt with the question of death of micronucleus containing cells by apoptosis, and they assessed a large number of cells, and they found that from the normal cells, less cells go into apoptosis than from the micronucleated cells. More of them go into apoptosis. So it seems that the presence of a micronucleus is a signal for the cell to go into apoptosis, although not all of the cells do it, but some do, more than in control. And this is about chromotrypsis, and this is really an interesting process. I just have briefly mentioned it before. Now let's look at it again. You have the normal cell, you have the micronucleus, and you might get a correct integration. If, for example, there's a whole chromosome, it still has a centromere, it's able to reattach to the spindle, and it reintegrates correctly, theoretically. Now, if you have this situation, you get a fragmentation of micronuclear DNA. You have intact chromosomes from the nucleus, and you have this fragmented DNA. But this fragmented DNA rearranges, sometimes at least, it rearranges into a chromosome that's totally out of order, that's totally not a normal chromosome. And that can be reintegrated into the main nucleus, and that's called chromotrypsis, and that's definitely a mutated cell. You could ask, is it different between different cell lines? For example, rodent cells, which have a less strict control against malignant transformation, or stem cells maybe that have a very strict control also against mutations. For rodent cells, there's really no study available to compare them directly to human cells with this background. For human cells, the genomic integrity, it might not be controlled so much if you have a differentiated cells, because if you have a differentiated cell, it has its task in the body, or it dies off. Think of a skin cell or something, it has its task, and then when it's done, it dies off and goes away. But a stem cell, that's a cell that still divides, and if it's mutated, it can cause a tumor, or be the first cell of a tumor formation. And so the cell has to control, the body has to control very strictly the quality of the DNA. So maybe there's a limited possibility for micronucleus formation in stem cells. We tried that with hematopoietic stem cells, which is shown on the left. This is doxorubicin doing the damage, and these are TK6 cells. And we found that you can induce micronuclei quite well in these hematopoietic stem cells, at least. You can induce them quite well, but they were more resistant than TK6 cells. So there is a difference between cell types, but it's not that the stem cells can never get any micronuclei. So the conclusion of the working group really, who searched all the literature available at this time point, which is already a few years ago again. Now I guess we could do another work group and another review, but we concluded then that primary cells control DNA integrity rather tightly, and if you have cells with a large damage, they're not permitted to survive. However, initiated cells on their way to tumor formation lose this control, and micronucleus formation may then be an aspect of genomic instability, which may enable further steps in the development of malignancy. So we think, or this working group had this conclusion, that genomic mutations such as micronuclei are relevant for cancer risk. It is an endpoint which really tells us about cancer risk, something about cancer risk, and not just dying cells. So it can be assumed with some confidence that micronucleus formation can really contribute to malignant cell transformation. And this assumption was really further supported by the papers talking about chromotripsis, which have appeared in the last few years. Okay, how are we in time? Do I have a little bit more time? Or would you rather want to ask questions? Because I could skip the next slides, but so we'll just go on and finish. Okay. So micronuclei and ecotoxicology, and how can you use this endpoint to do ecotoxicological investigations? Micronuclei are always for genotoxicity, for genomic damage, but that part of it can be assessed even in ecotoxicology. So what can you do? You can just take a test compound or a test whatever item with environmental concern and test it in mammalian cells, just like a normal in vitro micronucleus test. Or you can use plants or animal species to monitor genomic damage in wildlife. Or you can use animals or plant species which represent an environmental exposure. You can use them in experiments, in exposure experiments. And I have just a couple of examples for each of these possibilities. For example, you can test wastewater. I was involved in a micronucleus study doing that. We had a round-robin study. They gave us samples, coded samples of wastewater, and they spiked it with compounds to see whether all the labs that were involved would find a similar result. And it worked quite well. So the wastewater spiked with the compounds. It was detected as being positive. And here it was a more extensive dose response with this compound, and it worked quite well. So it is a robust and practicable method for the testing of genotoxicity of wastewater samples. Again, some more examples for other things like using animals and plants for experiments. For example, you can use the rainbow trout, and there you use renal erythrocytes. So you cut a little bit of the head of the kidney, and you just smear it on a slide. And you count the erythrocytes for micronuclei, And what they found in this particular paper is that after intraperitoneal injection into the trout, you get these results, a positive response with some typical positive control test compounds. So you can use rainbow trouts in an aquarium, in a tank. This is another example of fish that were injected with mercury chloride solutions intraperitoneally, and here they took the blood via this caudal vein puncture, and they analyzed the erythrocytes, and this is what it looks like, and they got a dose response. And here we have another example. It's butterflies here, and the reason for that was that they have this certain butterfly, this name, and it's a protected butterfly. It's declining in Europe, but it really is very present in rice fields in Italy. So they figured that the agrochemicals that are used on rice fields could threaten the survival of the butterflies, and they wanted to see whether genomic damage is also an issue here. So they tested some herbicides on it, for example, glyphosate, and extracted cells from larvae, and they sprayed them with a solution, and then they looked at them, and they found significant differences between glyphosate exposed and control cells in terms of micronuclei. They say that for the first time in their paper, a micronucleus assay was used to evaluate the genomic damage in such a invertebrate at the larval stage. You can also do biomonitoring of wildlife, and just want to give you some very brief examples for that as well. For example, here you can use muscles. You use the blood cells, they're called hemocytes in the muscle, and you can either treat them with genotoxins at a defined concentration, but you can also look at muscles from different field locations that are more or less polluted. And they found that the muscles collected from 2 polluted sites showed an elevated micronucleus frequency, indicating the presence of genotoxic pollution. You can also look at wild birds. This is a study from Brazil, and since they're birds, the erythrocytes again have nuclei, and they thought that these birds are very good bioindicators of the natural environment. They investigated birds from agricultural areas in relation to conserved areas. And again, they found an increase of micronuclei in the more polluted areas. And then there's this paper using bats. They captured 35 bats from 3 different colonies in Mexico, and they categorized the pesticide exposure of these sites as low, intermediate, and high. This is a little bat. It weighs about 10 to 20 grams, but it seems to be very distributed throughout Mexico and also other countries. They took 2 microliters of blood from them and smeared them on microscope slides and counted them, and they found an increase of micronuclei in the more polluted areas compared to the control area. They say this is also a useful marker for evaluating risk of exposure in wildlife populations. You can also use plants. This is a paper which investigates the toxicity of landfill leachates, and there's zebrafish, but there's also in this paper, but there are also mung beans. You can use the seedlings of these, and this is a mitotic index. This is a micronucleus frequency. You can use the root tips because the roots grow fast. You have a lot of mitotic activity in there, and you can then analyze the resulting micronuclei very well. And this is the second to last slide, I think. And this again shows plants and micronuclei in plants. And here they've used paracetamol to see whether the plants are a good model for experimental environmental observations of genomic damage. And you can see the mitosis very well and mitotic disturbances in these cells very well. Also here, chromatin bridge. And this is what the micronucleus looks like in this staining in these cells. And you can see with increasing paracetamol concentrations, that you get an increased micronucleus frequency. So the overall conclusion from this whole presentation is micronuclei are an excellent and also easy to assess endpoint for chromosomal and genomic mutations in biomonitoring and in genotoxicity testing, as well as ecotoxicology. It's very versatile. You can use it for many different purposes. And it is becoming more clear that the micronucleated cell itself can be involved in the carcinogen process. So it is a relevant endpoint. And also a deeper knowledge about the fate of micronuclei and micronucleated cells might in the future help to understand the processes of chemical carcinogenesis even better and better. Okay, thank you for listening to my presentation.